Hi, my name is Ana Uruena, and this is my presentation on ErbB-2 receptor transport. So for experiment 1, it involved RNA interference, or siRNA, in which the objective was to analyze the levels of important B1 in the cells and presence of ErbB-2 in the cytoplasmic and nuclear fractions after being subjected to RNA interference, siRNA. So this allows the analysis of relationship and effect between important B1 and ErbB-2 protein transport from the cytoplasm to the nucleus. The siRNA and immunoblotting allows you to use antibodies against each of these proteins in order to directly quantify siRNA and the relationship with important B1 levels. This allows for one to see the ErbB-2 nuclear input requirements. So the top panel for the figure 1 is the important B1 antibody. And based on the results, one can see that the levels of important B1 were present in cells and not subjected to siRNA in cells subjected to non-sequenced siRNA. So you can conclude that they were reduced for those with expression of important B1 in both cytoplasmic and nuclear fractions, but more so in nuclear fractions. In the bottom panel is the ErbB-2 antibody. It allows you to see that there is a high presence of the ErbB-2 in the cytoplasmic fraction of all cells, but low presence in the nuclear fraction of cells. Within the nuclear fraction, you can see that the ErbB-2 expression is somewhat proportionate with the important B1 panel, as the least presence of ErbB-2 was also noticed within the cell that has a reduced expression of import B1. So in experiment 2, it involved immunoprecipitate examination. And the objective for this experiment, which is in regards to figure 2, is to immunoprecipitate lysates of cells expressing either wild type ErbB-2 or mutant ErbB-2 lacking nuclear localization signals with antibodies to ErbB-2 or a control antibody. With this, you can examine them by immunoblotting with antibodies to important B1 levels or to ErbB-2 and see the nuclear translocation relationship within them. So the conclusion for this experiment, based on the results from figure 2, one can see that the control antibody MIGG did not associate or show presence in either the wild type ErbB-2 or the mutants ErbB-2 for either important B1 or ErbB-2 panels. In the important B1 panel, with the results, it allows you to conclude that the nuclear localization signal is in fact required within the important B1 for both antibodies. The wild type ErbB-2 showed ErbB-2 antibody binding, but not for the control antibody. For the ErbB-2 panel, with the results to the right, you can conclude that the nuclear localization signal shows binding within the ErbB-2. And the wild type does too. So it displays this membrane signaling pathway still works, but not with the control antibody. So there are alternative experimental methods that you can use. And another experimental method that researchers may have conducted in order to investigate protein-protein interactions includes fluorescence resonance energy transfer, also referred to as FRET. Identifying key players and their interactions is fundamental for understanding biochemical mechanisms at the molecular level. There are many optimal techniques available, FRET being one of them. There are properties to consider when designing a PPI analysis in order to determine which method is more suitable. Figure 3 gives an idea of some pros and cons for various methods that are available. FRET is a powerful technique for studying molecular interactions inside living cells. It is a distance-dependent physical process by which energy is transferred, non-radiative, from an excited molecular fluorophore to another fluorophore by means of intermolecular long-range dipole-dipole coupling. These donor-acceptor fluorophores come from a class of autofluorescent proteins that are known as fluorescent markers called green fluorescent proteins, or GFPs, which tracks proteins in living cells, allowing monitoring within the cell. So transport through nuclear pore complex. As illustrated in figure 4, this involves transportation of proteins made in the cytosol for use in the nucleus containing a nuclear localization sequence, NLA, for transport through nuclear pores. Nuclear pores are actually gatekeepers of two-way traffic. So to understand the figure, you kind of have to go through the steps 1 through 5 and understand the way in which this transport through the nuclear pore complex takes place, which is explained in figure 4, or also labeled 16.33 from the textbook. The transport actually involves the roles of various factors such as RAN or RAN. It involves GDP and GTP, important and GEF. An RAN or a RAN is an important molecule that is a GTP binding nuclear protein that is encoded by the RAN gene. This interaction between important and RAN can cause the NLS containing protein to be released for use in the nucleus, as seen on step 3 from the figure 4. The GDP and GTP involved in the binding of important and provides energy source. The GTP hydrolysis step provides the energy from nuclear import, where nuclear transport can be inhibited by exposing cells to non-hydrolyzable analogs of GTP. Important is a special receptor protein which binds to the NLS and mediates the movement of the NLS containing protein to a nuclear pore. And the GEF, these are actually proteins that stimulate the release of GDP to allow binding of GTP. So it maintains high levels of RAN GTP concentration. Now in figure 5, it's the assessment by immunoblotting for the presence of ErbB-2, important B1 or beta 1, tubulin, and histone H3. One can see that the cells containing RAN Q69L reduce the expression of the ErbB-2 in the nucleus, unlike the cells induced with the wild type RAN or untreated cells, which capped ErbB-2 expression. The RAN Q69L indicates the reduction of nuclear localization of important B1. A mutant that is dominantly negative, like RAN Q69L, and lacks the GTPase activity, prevents nuclear import of the protein within the cell. The GTPase activity of RAN is required for the transport of RrbB2 from the cytoplasm and nucleus. This indicated that RAN activity is required for the ErbB2 transport to take place. So a model proposal that explains mechanisms by which ErbB2 is translocated into the nucleus includes the forming of a complex which transports to the nucleus by the nuclear pore complex, or NLS. As one understands, ErbB2 requires important B1 in order to be transported to the nucleus. This transport must be facilitated through the presence of NLS tagging. These tags proteins for import into the cell nucleus by nuclear transport and allows you to see the association of ErbB2 release. The use of RAN GPPNP instead of using RAN Q69L would actually cause results to have a higher ErbB2 and important B1 expressed within them. As RAN GPPNP is actually a non-hydrolyzable form of RAN localized in the nucleus, this leads to the important B1 and ErbB2 complex binding to take place in the nucleus. So the results between the two will differ as the important B1 and the ErbB2 will decline in the cytoplasm. And there will be a variation in the energy requirements between the two, as it will vary by the nucleoplasmic events. And that is the end of my presentation. Thank you for listening.